Elucidating the molecular architecture of biological systems requires precise interrogation of protein dynamics. This is a domain of co-immunoprecipitation, coupled with mass spectrometry, Co-IPMS, a pivotal tool in functional proteomics. Within the cellular milieu, biological function is rarely executed by isolated monomers. Instead, proteins assemble into stable or transient macromolecular complexes to drive signal transduction and metabolic regulation. Deciphering this interactome is essential for a mechanistic understanding of cellular physiology. Historically, characterizing these complexes presented a significant methodological bottleneck. Conventional affinity tools offered limited resolution, restricting researchers to a fragmented, low-dimensional view of cellular networks. This created an analytical dichotomy. Traditional methods, like Western blotting, are inherently hypothesis-driven, requiring a priori knowledge of the specific interactor to select detection antibodies. This validates only the known. Conversely, systems biology demands unbiased discovery, the ability to characterize the complete local environment of a target protein without prior assumptions regarding its binding partners. Modern proteomics bridges this gap by integrating specificity with discovery. This methodology fuses the stringent selectivity of immunological capture with the high-throughput analytical power of mass spectrometry. The Co-IPMS platform represents a synergistic combination. First, co-immunoprecipitation serves as the affinity purification step, isolating the target bait protein along with its associated prey complex from crude lysate. Second, liquid chromatography mass spectrometry acts as a detector, providing sensitive, label-free identification of all co-purifying species. Optimizing the workflow is critical for data integrity. The transition from a physiological environment to a high-confidence interaction dataset involves a rigorous, multi-stage protocol. The process begins with cell lysis under non-denaturing conditions. Specific antibodies capture the bait-prey complex. Stringent washing removes nonspecific background contaminants while retaining physiological interactors. The eluted complexes undergo enzymatic digestion, typically with trypsin, generating a peptide mixture. These peptides are analyzed via tandem mass spectrometry, measuring mass-to-charge ratios with ppm-level accuracy. Finally, bioinformatics algorithms filter false positives to reconstruct a high-confidence interaction map. This workflow offers distinct advantages over legacy assays. Regarding approach, we transition from targeted confirmation to hypothesis-free discovery. Regarding scope, we simultaneously identify hundreds of potential partners, including isoforms. And crucially, regarding output, the data is quantitative. We determine the relative abundance of interactors, providing stoichiometric context rather than a simple binary result. A defining capability of Co-IPMS is its sensitivity toward transient interactions. Many regulatory events and signaling pathways are characterized by weak affinity or rapid turnover. While often missed by traditional blotting, the high dynamic range of modern MS instruments allows for the robust detection of these low-abundance, fleeting partners. This technological leap shifts the output from raw data to biological context, enabling researchers to interrogate the functional consequences of protein networking within a specific experimental system. What is the translational value? A quantitative map of the protein interactome allows for the reconstruction of pathway perturbations under varying physiological states. This provides a systems-level perspective. Beyond a simple parts list, we observe the remodeling of molecular machines during disease progression. We can quantify how a therapeutic candidate rewires interaction networks in real time. This is critical for identifying upstream regulators and central hubs in signaling cascades. Consequently, Co-IPMS has become a cornerstone technique across the biomedical spectrum. Its implementation is driving mechanistic breakthroughs in high-impact fields, including oncology, neurobiology, and infectious disease research. In oncology, we map aberrant signaling complexes driven by oncogenes to identify novel druggable targets. In neurodegeneration, it characterizes the interactome of misfolded proteins, elucidating aggregation pathways in Alzheimer's pathology. In virology, Co-IPMS maps the host-pathogen interface, revealing how viral proteins hijack cellular machinery, knowledge that is foundational for antiviral design. In summary, Co-IPMS represents more than an incremental improvement. It is a paradigm shift in how we approach functional proteomics. The analytical focus has evolved. We have moved beyond the isolation of individual proteins to characterization of integrated networks. Function is an emergent property of the network, not the isolated component. By combining the stringent selectivity of antibody-based capture with the analytical depth of mass spectrometry, we are effectively decoding the language of cellular communication, moving from qualitative observation to mechanistic understanding. The human interactome remains largely uncharted territory. Creative Proteomics is ready to help you map these hidden networks and accelerate your discovery.